Ladies and gentlemen, very good morning to one and all gathered here. I give me an immense pleasure to welcome you all to Inspire Internship Camp supported by Department of Science and Technology. Each new day brings new hope for a fresh start. A great momentum starts with a simple thought in the mind. It begins with a little step forward. With these wishes, let us count our days with golden hearts. I welcome you all to this great occasion.
During the 11th plan period, the Department of Science and Technology, Government of India, initiated an ambitious program, Innovation in Science Pursuit for Inspired Research. The Inspire program aims to attract bright young minds to a research-based career in engineering, medicine, science and technology. There are various programs conducted by the Department of Science and Technology like Inspire Awards, Inspire Internship Camps, Inspire Scholarship for Higher Education, Inspire Fellowship and Inspire Faculty Scheme. Among these, Inspire Internship Camp is a scheme intended for students who are within the top first cutoff in the 10th standard examination of any state education board. 50,000 children attend these camps every year. The camps bring students in contact with scientists so that they discover the excitement of doing science. The aim of this camp is to motivate them to use scientific thought process in all walks of their life and to inculcate scientific temper. It is our privilege to saying that this Inspire Internship Camp was conducted by 6th time in this institution.
Prayer and seed of faith are similar to nature. Both have nothing within but have the potential of create everything, and they make impossible things to possible. Let us all stand for Tamil Thai Valtu and give a good atmosphere and auspicious beginning to our day.
Thank you. As the light from the sun which falls on the flower that will make bloom and give fragrance with happiness. As we work to create light for others, we naturally light our own way. Now I request our college President, Engineer Pon Balasubramanian, Secretary Tirupon Ravichandran and Principal and our today's Chief Guest Dr. M Palaniandavar to share their hands in lighting the Kuttu Velakku and give auspicious talking to this day.
Come, one of you, any one of you. No, madam, very good, excellent. Grab the opportunity.
Thank you, sir. Leaders have become great not because of their power, but because of their ability to empower others. Now I welcome our college Principal Dr. A.R. Pon Periyasamy to welcome the guests and the gathering.
Very good morning to everyone gathered here.
Good morning.
Honorable Chairman of our college, most respected President and Secretary of our college, today's Chief Guest, Dr. M Palaniandavar, Professor of Eminence and INSA Honorary Scientist, former Principal Dr. Jayaprakasam, Coordinator of the Inspire Camp as well as Coordinator of Self-Finance Program, Dr. Meenanchi Sundaram, faculties of our college and other institutions, students from various schools. Good morning to everyone gathered here. Good morning to one and all. It gives me a great pleasure to welcome the dignitaries of the dais and of the dais, guests and invitees, and my dear colleagues and students, I welcome all of you for the 5 days Inspire Camp. I am very happy to welcome our dynamic President, Engineer M Palaniandavar sir, to felicitate this function. He is an engineer by education and is a very good administrator and present taking care of all the things in the campus. He has been very helpful and leading us right direction in our department in next higher level. On behalf of the participants, I extend a warm welcome you, sir. I am very glad to welcome our college secretary from Ravichandran, who is always an inspiration for each one of us. It is my honor to welcome him to the Inspire Camp. Welcome you, sir. I feel proud to welcome Dr. M Palaniandavar, Professor of Eminence and INSA, for accepting to grace this occasion and to deliver the inaugural address of this science camp. Professor started his academic career in Madurai Kamarajar University as lecturer in inorganic chemistry and then became reader in inorganic chemistry. In 1986, he moved to Baradas University as Professor of Chemistry. A total of 18 research projects sponsored by DST, UGC, etc. He has guided 27 PhD. 180 reviewed research articles. He served as member DST PAC on inorganic chemistry during 2002 to 2007 and then as member in committee DST fast track young scientist scheme. He serves as expert in INSA, Inspire, UGC, etc. It is my honor to welcome him for the Inspire program. Welcome you, sir. Hearty welcome to former Principal Dr. Jayaprakasam, coordinator, and participants from various places for attending this Inspire camp. Special welcome to all faculties from other departments of our college and various schools for taking the time from their busy schedule to grace this function. Once again, I welcome all of you for the inaugural function of the science camp under the Inspire internship scheme. Welcome you all. Thank you.
It's not our purpose to become each other. It is to recognize each other, to learn, see the other and honor him for what he is. Now I request our college president to honor our chief guest, Dr. M Palaniandavar, Professor of Eminence, INSA Honorary Scientist, Department of Chemistry, Bharathasan University, Tiruchirappalli. Thank you, sir. Action, inspire others to dream more, do more and become more. Those are become achievers. The above said words highly goes with the person who is none other than our college president, Engineer Pon Balasubramanian, whom I am called upon to deliver the presidential address. I am delighted to offer a happy welcome you, sir.
Good morning.
Good morning, sir.
Very tired already. Having travelled all along. Okay, let's hope this 4 days in 5 days Inspire camp will regenerate you all. First, congratulations to all the participants.
Thank you, sir.
Because you have already proved that you are going to be a successful person by securing 471 marks in your 10th standard. Vilayum Payir? So, select karma because of your high score and your interest and enthusiasm. Now, the government has come forward to motivate you to do research in science. A lot of things have happened. The humanity has developed because of research in pure science, be it the electricity, be it the telephone, you name it. The humanity has developed only because of the science research. So the government wants to encourage you, motivate you to do research in science. So that is the very purpose of this camp, 5 days camp. I know many of you have skipped your quarterly exams and you have come here. I must appreciate at this point the managements of the schools who have accepted to send the students in spite of the exams. I am sure these 5 days will transform you and your outlook to be a successful scientist in the world, innovating better things, making the human mankind to live in a better world. Wish you all the very best.
Thank you, sir.
So, chief guest of today's function is none other than our Dr. Palaniandavar, who served Bharatdasan University for quite many years as HOD of Chemistry Department, and he has also served, as Principal said, he had been in very many committees and he has done a lot of research in chemistry. I am sure with his inaugural address, you will start your journey to become a scientist in the years to come. Thank you, sir, for accepting to be here, and we are really honored to have you here. Not only Dr. Palaniandavar, there are going to be a list of other scientists who are going to inspire you why you should take up science as your career. I am sure this 5 days workshop will give you that thought process. Generally, the parents will say, okay, you have to become a doctor, you have to become a science engineer. So, only these two professions are imbibed in our minds, thinking that these are all the two careers we should opt for. So, to change that mindset, this 5-day workshop is being organized. I am sure you will have one more option at the end of this camp, whether to take up as a scientist or not. So, I leave it to you. You can decide. You are all, people who can make decisions of your own. Okay? So, after this 5 days workshop, you can decide whether you want to be a scientist or an engineer or a doctor or whatever it may be. So, that is the very purpose, and I must congratulate our principal, former principal, and the Inspire Camp coordinator, Dr. Minat Sundaram, for making elaborate arrangements. There can be many shortfalls. Please accept, because you are here to get yourself inspired. Okay, this is a remote rural village school, college. So you may have some difficulties. Some of you could have come from very well-to-do institutions where you might have a lot of facilities. But here we have bare minimal facilities. I am sure you will enjoy it, and it will help you to live. When you have very minimal facilities, how to take your life forward, you can learn from this institution. Wish you all the best, and God bless. Thank you.
Thank you.
Excellence is going ordinary things extraordinary well. Here we have such an excellence person who is none other than our college secretary, Tirupon Ravichandran. Now I welcome him to felicitate the gathering. Welcome you, sir.
corresponding future scientists? That's nice. So, the 5-day Inspire program is going to transform you all to be the next generation scientists. See, if you talk about engineering or medicine or anything, it's just application of science. Whereas the future scientists, you are all going to create something new. Whatever you create has to touch the entire humanity. For example, in the olden days when there was no matchstick, people were using stones to light fire. When matchbox was introduced, how it transformed. Imagine the amount of transformation it brought in, the happiness it brought in the entire mankind. It made life easier. So as scientists, that is what is going to be your job. Your job is going to find a better method, new technologies, new ideas to make life simpler for the common man. As a scientist, you will all be given this task, and whatever you do, when you achieve, you will be touching the lives of very many people in the whole earth, because any new development, any new research finding is going to help the very many people to make their life simpler, easier and better. So that way you are all going to be the agents of change. You are all going to be the agents of change and transforming the people's lifestyles into a simple and easier way of living. Okay? So with that, I am really happy to invite you all to our institution. As our president said, enjoy your stay here for the next five days, and you will get transformed as such, and all the very best. I would like to take this opportunity to thank Dr. Palaniandavar, the professor and scientist of INSA Honorary Secretary, for taking time off and being here for the inauguration session. I would like to thank our principal, our president Mr. Balasubramanian, our former principal Mr. Jayaprakasam, for conducting the 6th Inspire program. I would also like to congratulate Mr. Meenakshi Sundaram for continuously conducting successful camps as the 6th camp this year. I would like to congratulate all the faculty and other staff members here who have been part and parcel of our institution. Thank you very much. Wish you all the very best. Good luck to you.
Thank you, sir. The scientific man does not aim at an immediate result. He doesn't expect that his advanced idea will be readily taken up, but he gives a lay foundation of those who are to come and point the way. Here we can such a scientific man with us, who is our today's chief guest.
Dr. M Palaniandavar, Professor of Eminence and INSA Honorary Scientist, Department of Chemistry, Bharathasan University. Sir's area of interest is bioorganic chemistry and takes major contributions to the field of structural functional models of electron transfer of copper proteins. He is the only person in India to initiate the research work on metal-DNA interaction. He founded a Center for Bio-Inorganic Chemistry, and he served as a distinguished visiting professor of chemistry at IITB Bombay. He was elected as a fellow of the Indian National Science Academy and Academy of Sciences and Royal Society of Chemistry. He received the Tamil Nadu Scientist Award, UGC Research Award and Professor Sankey Gowda Research Award. He served as a member in CSIR NET Committee on Chemistry, expert in INSA Inspire, UGC committees, and also member in the editorial board of many national and international journals. He has organized several winter schools, refresher courses, and academic lecture workshops in many universities and colleges for the benefit of students and scholars. Sir received many projects from various sectors like DST, Nano Mission, CSIR, BRNS, and UGC. Sir has published more than 180 research papers and guided 27 PhD scholars. Sir was awarded Japanese Science Promotion Society long-term visiting professorship and DST Japan Science Promotion Society invitation fellowship and visited many universities in Germany. It is our pride to have such an eminent scientist admit with us, and I welcome you to deliver the inaugural address, sir. Welcome, sir.
Very good morning to all of you at the outset. First, I should also thank the management, Engineer M Palaniandavar, M Ravichandran, the management of this college, famous college, Principal Dr. Puthanampatti, and also your coordinator of this program, Dr. Meenakshi. I thank them for giving this opportunity of addressing the young minds. Right? And the most important thing here is to think about the Inspire program itself. They read about the Inspire program, but people forget about the origin of this Inspire program. This program was conceived by Dr. T. Ramasamy, who was the secretary of the DST, was formerly director of the Central Leather Research Institute in Chennai. So he has been my good friend and colleague, and whenever we meet him, he used to talk about this program. And this was facilitated by none other than Bharat Ratna Professor C.N.R. Rao, who was then the Prime Minister's Scientific Council Chairman. So a lot of people are there, big people are there behind this particular movement. In fact, I should tell you, this Professor Rao, who came to Bhathidas University long back when I was heading the department, and that's called Jawaharlal Nehru Center Lecture Series. He organized lecture series long back. He came to our Bhathidas University, addressed our youngsters. What is science, what is chemistry, what we should do like that. And that is the origin of all these activities. He found not only happiness in that, organizing that, and he found that that's a very good result of this one. Young students who have not visited Indian Institute of Science, where he worked. Jawaharlal Nehru Center for, Jawaharlal Nehru, JNCSR, Jawaharlal Nehru Center for Advanced Scientific Research. He only, founded that as a deemed university now. Very advanced research institute. So he found that such types of meetings always help people, students like you, to know about what is going on around you. That's the first thing. Then there is an opportunity for him to tell you about what is going on in science. In fact, I should tell you, he has established a science museum in Jawaharlal Nehru Center for Advanced Scientific Research in Bangalore. Right? So when you have an opportunity, visit that. So he wanted to create, a flavor for science. In fact, another time, 2006 or so, I brought him here to our department. He organized an academy meeting, annual meeting, this Science Academy, that's Indian Academy of Sciences, founded by Sir C.V. Raman. That was conducted in our chemistry department, chemistry and physics department in Bhathidas University. At that time, he organized conferences. I also requested him to address students like you. He addressed a big gathering of 2,000 students in BHM. Right? I didn't tell him initially that he will be addressing the students, but later on he told me, Palani, you should have, he addressed his main name very well, Palaniandavar, like that. So he told me, Palani, you should have told me that I am addressing the students. And I would have prepared a different lecture. He's a very enthusiastic person. He's more than 84, 85 or so. But then his interest in science is not, below everybody. He's Bharat Ratna. Bharat Ratna. So it's still it is there. He's very busy nowadays. Whenever I go to see him, you have to wait and see him. He's very busy in his own science, doing a lot of material science research. Right? So he wants to, he was, people say whenever I travel Germany, people there used to process there, used to tell me that next Nobel Prize will be for Professor C.N.R. Rao. That is, he had a lot of discoveries in the so-called new materials, particularly the carbon nanomaterials. Right? So, but then he missed it. So that doesn't mean that he is not interested in science, right? His interest, whatever he invested in science, is not wasted. Now you see a lot of institutes have come up, particularly for science. I think you have heard about it. Indian Institute of Science, Education and Research. He initiated it. Right? There are five such institutes, ISIS. You just go through your website, you just go to these institutes, they have grown up very well. Right? And they take up faculty members directly from abroad. They do very good science. Nowadays, universities, people say, are dying. And these people, when there is one in Trivandrum, one in Kolkata, one in Bhopal, there is one in now they have started one in Tirupati, right? One in also in Mohali, right? There are a lot of institutes. To tell you the truth, the directors of these institutes, they visited us, the chemistry department in Bhagirath University. We were doing very well, Bhagirath University chemistry department. We had several such people, including Ramaswamy and all. They all visited our department. Right? They just motivated us. So these, directors of these institutes, they are none other than famous chemists. They are all my good friends. So I wanted to tell you this one because you should also know what would be the ultimate aim of taking up science research. Right? Apart from Indian Institute of Science in Bangalore, where I was there last week to give a lecture, these institutes are coming up here. There is also one NICER, National Institute for Science Education and Research, that was funded by BRC, Bob Atomic Energy Commission. That's also headed by my own good friend, Professor T.K. Chandrasekhar, Professor V. Chandrasekhar. They all see to that young people like you are attracted to this. They have 4 years course. Okay, they have a 4-year integrated science course. And such an integrated course is there in Central University Tiruvallur also, where I worked. Right? So all these courses aim at very talented people like you. You should go there and see what science means. What are all the opportunities you have. You will learn science, real science there. So this I am telling you because I should also tell you, not only to motivate you to take up science, but I should also tell you about the opportunities available. As he said, once you finish this program, and you can get very inspired if you stand first, first in a college or in a school like that, you will get an opportunity, scholarship, to do PhD, integrated science PhDs there, in all these institutes. So the very institute started by these people, Professor Rao, right? And several central universities, 15 central universities, right? They all aim at taking science up. So there are several opportunities. You should try to apply to this and get benefited. So science aims at not only this type of your good career. Professor Rao used to tell me, people who take up IT, right? IT people, they used to tell me very jovially, they are foolish like that. Every day they do some routine work without any spike in their life, right? I've also seen a lot of IT people coming to take up agriculture, right? So this science, on the other hand, is very innovative, and it also gives you enthusiasm to discover something new, right? And you are encouraged to keep yourself young, right? There are a lot of opportunities, a lot of the good things about science, right? When you do science, you feel happy, and there are a lot of examples I can quote. A lot of people, when I give the lecture, I shall be telling you more about such people in chemistry who have enjoyed doing science, who have enjoyed talking to people like you. I don't think that if you do this particular this course or do this take up science and also visit various institutes of science, don't think that you are compelled to take up only science. Like Inspire, there is a program, SEED, SEED program, seeding program in the US. And I was reading an article the other day that an Indian origin student who did this SEED, who underwent the SEED program in a research institution, he took up accountancy. But then he says, this SEED program only gave him the enthusiasm. Right? So they made him think. Right? So remember, even if you don't take up science, this type of camp would help you Now taking science, thinking in terms of science, right? That will help you to make your life simple, make your life better. So science doesn't aim at only jobs and other things, but it aims at the fulfillment of your life itself. I should also tell you, in fact, I was noted down. I was reading an article again on the science education in India, right? And in fact, he says that I didn't know about this. Science in the Constitution of India itself is based on that. It says our society is science-based, right? And I was noted down some of the words. It says the science-based society is constitutive feature. It's there in you. It should alleviate ignorance in you. It should alleviate illiteracy in you. It should remove penury. So these are all the aims of taking up science education, not only getting a job. And the attributes of science, the good things about science is that you think rationally. You think rationally. So Tamil solralliya, eppol yaar kettum appol mei porul kaanga theriyum. Adhe pola science. They say there is something, you should think about it. You should ask why, who, everything you should ask. So that is how this camp also helps you. You start thinking science-based. You go to the US, I've lived in the US, right? So whenever I visit it, I find that people are mostly science-based. They think in terms of science. So our constitution is, right? Our country itself aims at that type of society, science-based, right? And growth of the individual, your individual, right? And also the society. Everything depends on science. So it should be science-based. When you read science book, I think I remember seeing, what is science after all? What is science? Science is, what is science? What it means? Simply, you say science and go away. What does it mean? Study. Science is, observation. Science is observation. And then you record your observation for your posterity. Right? So science means observation. Think. If you see, how this bird is flying, then what do? You try to make up your own equipment to fly. Right? So that is science. Right? Bible says, sakal kandupidichavanga unmiyana scientist. Right? However, science should form a part and parcel of your life. You should change others also, as the president of the college said. That should change you. Attending this meeting should change you. So I have participated in lectures in all these programs several times, but I always students come and talk to me, what is this, what is this, and that. And you should also notice there is one other program, KVPY. You know what is that? Oh, okay, good. You know that. So because we used to go and sit in IIT Kanpur, IIT Kharagpur and set questions for this. So whenever we set questions for, there are different levels, right? 3 levels. We are asked to set questions at BSc level. When I set questions for you, I was asked to set questions at the BSc first year level. Right? That means, just difficulty in this. You should also have a flair for science. You should read more, right? So that way, I tell you, it should be science-based. So it should form a science-based society, right? And you should also remember that when I just say science-based society, when this science developed in Europe, or how you have the basis of science, why I say it should be science-based. I read several articles, they always think about science-based society even in Europe. I have some articles on that. When did science begin? There are several stages. The first one is around 1600. That's when in Europe you get the so-called plague disease. You know what it is? Plague disease. So when I was an instructor, postdoc teaching and doing research in the US, when I went to my mentor university, Drexel University in Philadelphia, he had a book, plague disease. கேள்விப்பட்டிருக்கீங்களா? ஏரியால வருது கொஞ்சம் வருதா? Okay, blood disease. Okay, so that disease was all over there in Europe. Even now I read articles. How did they find out? You know, there are several techniques now. There are several books where they have maintained the names of people who died in this type of disease, blood disease. When you take the book, you can see in the old book and then you analyze the powders, particles there, there are techniques. Then you find that all those proteins there, no impurities, all those proteins are from the mice. Disease. So, other than protein and இவ்வளவு டெக்னிக் டெவலப் ஆயிடுது. அந்த டெக்னிக் வச்சு தான் அவங்க கண்டுபிடிச்சாங்க இந்த அந்த இடத்துல இந்த டிசீஸ் இஸ் பிளாக் டிசீஸ். ஹஸ்பண்ட்ஸ் deserting the wife, deserting the family, right? Children running away from parents. This was all there in 1600. That is when a real science developed. Okay, 1600. Then the next stage of science development, when? World War II. Lot of money was invested in, for example, in
postal எல்லாத்துக்கும் credit ஆயிடும்.
Money was invested, funds were invested in discovering atomic bomb in Manhattan, Manhattan project in New York, right? So a lot of money was invested in science. People realized that, for example, we talk about the Europeans, they ruled us, right? English people ruled us. What is that they brought? The first thing, toilet. Right? So this is the way they tried to change themselves, and they were also able to rule us. So science is the basis of scientific thinking, scientific discoveries. They should form the basis of our own society here. So remember, we should also try to do the same thing, and I should be willing to tell you another point. Wherever I go, I just find out another thing. If you go to Bengal, West Bengal, right? The textbook there, they have written for chemistry is different from what we have here. Right? Textbooks are different. Andhra Pradesh textbook is different from ours. Right? And our textbook, now I will tell you the few anecdotes. So it's different. So when we set questions for KVQY and NET exams, we find that their books are different. Particularly in West Bengal, now all of our people here, once we say engineering colleges, 550 engineering colleges, everybody rushes to that. But in Bengal it is not so. Half of the people, even 70% of the people, they want to do science. So whenever I sit in a meeting, right? Whenever I sit in a meeting in Delhi, I find most of them are Bengalis, my committee members. Most of these directors in these institutes, they are all Bengalis. So they are interested in science. Also they are interested in engineering, no problem. But most of them, they want to do very good science. Go to any ISER, right? Any IIT. What do you find? Most of them are Bengalis. In IIT also here, a few faculties now recruited, they are also Bengalis. Once when I visited IITM, out of the 28, 30, BTech, 30 MSc students in chemistry, they said, my colleagues there told me, 27 are Bengalis, only one is Tamil Nadu. So this all means that these people, Bengali people, have a good flavor for mathematics, physics, and then chemistry, and which is absent in our people, right? So we should try to seek good, institutions where you can have a good opportunity to do high-level scientific research, right? I mentioned some of them. Please remember them, right? And apart from, also this one, as I said, the syllabus you have, the books written there, they are not good. Once they tell you about some discovery, they simply say it is discovered, and then you read and then mug up and then go away. But they should tell you the history behind the discovery, right? Then only you will appreciate. In my lecture, I will be pointing out some of these aspects. So you should remember this history behind the science also. I should also tell you another important thing, that when you do a good science, you will be recognized. How? You will be, your seniors are there who will be addressing you, Professor Lakshmanan, Professor T.J. Pandian, who was my colleague in Madurai Commonwealth University. They are all Bhatnagar awardees. When you are 45, when you are young, people are 45, if you do very good science, you get Bhatnagar award. What is that? That is more or less equal to a Nobel Prize in India. Right? After that, you have, a good scientist is recognized by the fellowship. That's always I stress that whenever I write my name, put this FNA like that. What is this FNA?
Hello. Sir, இப்போ நீங்க
So FNA means Fellow of the Indian National Science Academy. How many people are there in chemistry? 110 people are there throughout this country. So you are recognized for this fellowship in science. Common Academy, that also gives you a fellowship, FASC. So these are all recognitions. You are consulted if you want to have some changes in the in science policies and all. So remember, there are several, apart from opportunities, I pointed out opportunities. You have good job, good salary, right? And in fact, my own student in IIT, he had 75,000 rupees as salary as such associate. Right? Apart from good salary, apart from good life, you also have fulfillment in life. You feel you are happy to do some science, right? Apart from this, you also have, recognitions by these academies. So in short, I should say, in India, you should try to take up science. Now people also talk about, say, no unemployment, but it is there. Once you are a selected person, you have a good job. At least 1 lakh to start with. You have a good job in science. I should tell you that 2 associate professors in IIT Mumbai, right, from my own department, my students, foreigner students. You go to any ISET, any IITs, you will find one person from chemistry department of Bharat University as MS student. They are all associate professors, assistant professors. You go to NICER, Bhubaneswar, I am a student as the chairman of the chemistry department. So we have generated very good people. We have invited a lot of scientists to our department, to our Bharat University, and we have trained our students very well. They all occupy very good positions in this country. This is a very good example how interaction with scientists. I brought a lot of people. My students interacted with them. So such an interaction will always help you. I've talked to people who attended this camp. They said this type of camp has encouraged them to take up science. They are all doing very well. From a rural area in Karur, I have my student who is Raja in IIT Kanpur, who did science with me with our chemistry department, who is an associate professor now. So remember, if you do well in science, right? If you invest in science, your efforts in science, right? You will be a very good leader in science, and you can enjoy doing science. So I wish that you also take up, you enjoy interacting with all the scientists. Try to see where they excel, right? Where they excel. How did they excel? By taking up science. You ask them, you interact with them, talk to them, right? Rather than just hearing the lectures. We will point out to you what are all the interesting aspects in science, right? Talk to them how they have come up in science, how they have been recognized, right? So I wish you all success in your life. Wish you all a very good time interacting with people, and I wish that you take up science ultimately. Thank you for your attention.
Now I request our Chief Guest, Dr. M Palaniandavar, Professor of Eminence and INSA Honorary Scientist, Department of Chemistry, Bharat University, to take over the session. Please, sir.
I think keep on talking English is fine for you? Okay? Good, right? Oh. Sometimes in some places I used to talk in Tamil also. It all depends on what you just try to get from me. Right? So what I will do in my lecture, right? I will try to talk about my own science, chemical science. I have noted down here, it is a central science. Why? Because chemistry is central to all science. Some people say mathematics is queen of science, it's fine. But this chemistry, the chemistry is not, I don't call it chemistry. It's a very small thing, chemistry. But chemical science is a very big one. Right? That's why I said it is a central science. I've also noted down here 2011. I prepared this slide long back. 2011, that is the International Year of Chemistry. What does it say? Our chemistry is our life and our future. Okay? So Professor Rao, I told you about, his efforts along the Royal Society. It's a very big recognition for science, right? Fellow of the Royal Society in London. Okay? So he used to say, the very big discovery in science will come only from chemistry. Right? So chemistry is like that. Not only that, it offers you opportunity, it offers you a lot of challenges. Everywhere you go outside, you see plastics. Right? But who has to do that? Solution. Give the solution. Chemist. You have to find out an enzyme, a catalyst, which can degrade all these polymers. Right? That is why I said, the chemical science is central to all science. It is also, an interdisciplinary science. What do you mean by that? Interdisciplinary science. Chemistry is the central science that responds to societal needs. That's what I'm telling you. When you have a problem, right? Polymers, plastics, everywhere, right? I have an article which tells you, the whole, just take the area of India, right? Go to the Indian Ocean, the same area of plastics are there on the sea. You understand what I'm saying? The amount of polymers and plastics dumped in Indian Ocean, that corresponds to the area of India itself. So much plastics are there. Take Sri Lanka, that has more plastics than India. Pakistan, more plastic than India. So, science, chemical science, is central science that responds to societal needs. Okay? So, you have a lot of opportunities. Has chemistry ended up? No. It has broadened itself. Right? It's broadening. Suppose you say this number, that number, Avogadro number like that. That doesn't end with studying about Avogadro number or quantum numbers. They are all basis for chemical science. Right? So, this science, chemical science, is expected to have a close involvement with society. That is the destiny of science. We have to interact with the people. We have to go to their help. Not only in producing, facilities for science, people. See, after all, what is the aim of science? Principles of science are converted into technology, engineering, right? What is the aim of science? It aims at increasing your life standard, a better life. The quality of life should be better. That is the aim of science. Convert scientific principles into technology and then help your life standard, improve your life standard. That's the science. When you do that, we invent plastics, but ultimately what happens? It spoils our environment, right? So we create, science creates, and that also requires some research activities on that. So remember, close involvement society is the destiny of science. Science is also beyond the science of mere observation and understanding nature. I told you, what is science? To study science without knowing the definition. It's aims of science, understanding nature. When you understand nature, you try to solve our own problems or betterment of life, right? So, chemists should understand other fields and also collaborate with other scientists. You should not be happy with doing science, chemistry, a reaction. the test tube. We should also understand what goes on in physics, what goes on in biology. So that is what I said. Nowadays the chemical science, chemistry is called chemical science, which is an interdisciplinary field. You should interact with them. That is what I am going to tell you today. What I did in my lab, I did inorganic chemistry, some inorganic compounds. And then I took it to the biologists. What are the biological importance of these compounds, right? So that type of thing is very important. Make it interdisciplinary. So don't think that chemistry is very small, or even physics is very small. They are all becoming interdisciplinary. And molecules, which are important in medicine, right? And cured of tens of millions of people of serious diseases and improved the quality of life. So that is the aim of science, remember. And what is the contribution of chemical science? How we improve in science, or how we get recognition in science? How do we generate money for the research? It should be relevant to the society. It should be applied, immediately relevant to the society. Now you understand what is science and how does it, what are all its aims, how we should have our research problem, that should answer the societal needs. So as I told you, when you write textbooks, you not just say principles, this is principle. You mug up and then write it in the exam. You should tell the story to future generations. For example, I tell you, vitamin B12, how it was invented. Why? Okay? This is what I will tell you today. So we should tell you the stories and also tell you about the opportunities of research and development for young scientists like you. Right? And I should also motivate you and young scientists to plunge into ever-expanding fields of science to overcome new hurdles. All what I said, plastic bag, that's a hurdle, right? So how to overcome that? These are all the aims, important tasks of science. The field of science is expanding. When our needs expand, science also expands. Okay? And science will continue to accomplish unimaginables. Okay? So long back, when you talk about chemistry in 1960 or so, people talked about very small chemistry. The definition for chemistry was very small. What is chemistry now? Can you tell me? You have read chemistry already. What is the definition? What does it do? What does it say? Chemistry. It's here. Make new compounds, synthesize them. Okay? And then you study their structure. How it is arranged. If it is sodium chloride, sodium plus is surrounded by chloride ions. Chloride ion is surrounded by sodium plus. That is the structure. Now, you also study the reactivity. If I take this wood, you think about what is its structure. And when you want to know the structure, with the help of the structure, you can explain the reactivity. When you explain the reactivity, you can try to see how you can use them. See, that is chemistry. That is, it deals with making the molecule, new molecules, determining the structure just to illustrate the dynamics. That is the reactivity. So if you know the structure of any molecule, any compound, any material in this world, you can illustrate their reactivity. So that should be our aim. Determine the structure, illustrate the reactivity, and also see how it can be used. Okay? Or simply you say, chemistry deals with formation of a bond, breaking of a bond. That is dynamics. So to illustrate that, should be there. So when you have studied about this one, you just expand modern chemistry. What is that? Linus Pauling formed the modern chemistry. Discovery of chemical bond. How a bond is made of electrons. Electrons, sharing of electrons. Remember that. There's a cooperation of two atoms through electrons. Okay? and the structure. Why you need structure? You want to explain the reactivity. Okay? And when you isolate new compounds, you have new disease, you have to isolate new compounds, right? You determine their reactivity, properties, etc. Right? And use spectroscopy, everything to explain that. And you should use the structure then to explain the reactivity. Right? That's what I have written. A good example is when you want to explain the properties or reactivity, you should know their structure. That is how a new science comes. That is what I am telling. Molecular biology. So when you want to know about our own function, how do we behave, then you should know the properties of all the molecules which have made up our body. So when you know the structure, you can just try to illustrate their properties. We say hemoglobin in blood. So you should know the structure of the hemoglobin. Then we say how does it interact with molecular oxygen and how does it carry efficiently from the lungs to the tissues. Right? So you should know the structure. That is what is the basis. When you know the structure, you illustrate that. When you study that, you say, can you have a small molecule, make a small molecule, a model for hemoglobin, right? And then see whether you can store oxygen. Take it in the bloodstream, use it in the tissue to burn our food. Our foods are burned, right, by oxygen, and then we get the energy, right? So that is how, when you take each and every molecule, every object in the world, you should know the structure. Then you determine their reactivity and their behavior. So remember, chemistry is an interdisciplinary subject. If you want to develop this particular field of how the DNA is there, how protein is there, how hemoglobin is there, you should know their structures. Then only you can understand all these biological functions. Right? So that's what I say, it's an interdisciplinary field. So modern chemistry, what are the new fields? Remember, people make up newer organic molecules. You have new disease, new problems, you have to make medicines. Okay? Make tablets, everything. And then you have to use new catalysts. What are catalysts? You know about this. Catalysts, right? Good. So you need catalyst. Right? So for example, what is photosynthesis? Carbon dioxide is reduced to carbohydrates. Have you ever thought about this? Can you make carbohydrate? Carbon dioxide is there. Have you ever imagined whether you can take carbon dioxide and make carbohydrate? In your laboratory, for example. In a test tube, for example. So you need to study photosynthesis. It is not a small one reduction. Carbon dioxide is reduced by the hydrogen atoms of water. Water is decomposed into hydrogen and oxygen. Oxygen is the byproduct. Hydrogen is used to reduce carbon dioxide to carbohydrate. Now all over the world, now people think about not only making carbohydrates, why not use carbon dioxide to make methane and use it as a fuel. Methane, it's a very good fuel. What is that you use in your house? Fuel. Low energy hydrocarbon. Low molecular weight hydrocarbons. Methane, ethane, propane, butane, like that. They only form your fuel in your house, right? So can you make them from carbon dioxide? You can. So when you observe something in nature like this, carbon dioxide reduced to carbohydrate, one can take up research to make methane or ethylene from carbon dioxide. That is what is going on all over the world now. People talk about energy materials, how to make them, new materials. Can you take a grass? The grass has stored carbohydrates in the grass itself. this thing. So what you can extract the carbohydrate, use it as a fuel directly, instead of burning them, instead of firing them, right? So these are all newer thinkings, okay? So remember, you can talk about newer catalyst. What is the catalyst for in photosynthesis? What is the catalyst? You cannot take carbon dioxide. Chlorophyll, good, magnesium containing a complex molecule, right? So that is a catalyst. So for any such reactions, you need to invent a catalyst. You can invent a new catalyst, right? That's what we have been doing in our laboratory. Burn food with oxygen to get carbon dioxide back and energy. So what is that I was doing in my laboratory? I was trying to make a catalyst which can burn organic compound, convert an organic compound into another one. So you need to use molecular oxygen. People talk about the use of sunlight. Same thing. There are secret meetings about in Europe how to use molecular oxygen. I think you have done a reaction. You have oxidized some organic compound in your laboratory, some oxidation reactions. Potassium permanganate, so high energy oxidizing agent. Why do you use high energy oxidizing agent when you have molecular oxygen around you? But see, we are also made up of organic compounds, but we are not burnt. Why? We need a catalyst. We are not burnt easily by molecular oxygen. Right? You have to invent a catalyst. So, molecular oxygen utilization, that itself is a very good separate chemistry I can go on talking about. Molecular oxygen. Right? So, a lot of newer fields are there, newer chemistry is there. So, you should also see about the green chemistry. See, it's called a responsible science, chemistry. It's a responsible science. You have to also tell people how to destroy the polymers. Right? Then you have the responsibility. And future of the civilized society, you should maintain that. So you have a lot of responsibility as a scientist. Just doesn't say fix A and B, you get a new color, new thing, and be happy and then go away. No. It's a responsible science. You should react to the society and help them to eradicate difficulties they come across. Right? And the future of chemistry also, nano and new materials. All these catalysts can be made of these nano materials, new materials. That's what I said. Professor Siena Rao has an advanced materials laboratory in Bangalore. Right? There are several institutes in India, nano chemistry institutes, nano materials institute. One in Bangalore, one in Mohali, and another one in somewhere else, I don't remember. So all these institutes have been formed to talk about these nano materials and new materials. Right? So instead of waiting for years to make a catalyst, you can make a small nano material and use it as a catalyst. So a lot of developments are there in science, particularly in chemistry. And then most important thing is energy. Nowadays people talk about what? Energy, right? So how do you get energy? Use all your petrol. Now people, you read in newspapers, people drive, I read in newspaper, use water as fuel. Can you use that? Water as a fuel? Can. I told you just now, carbon dioxide can be reduced with the hydrogen atoms of water. H2O is there. So you can decompose cut that water into hydrogen and oxygen. How can you do it? Take a test tube, put water, put 2 platinum electrodes and put a battery. It's a strong electricity. And then you will decompose into hydrogen and oxygen. But look at the leaf, green leaf. What's going on there? Photosynthesis. What is that you find? Water is decomposed at higher temperature. Leaves do that. Water is decomposed at a pH of 7. At ordinary temperature, you get hydrogen and oxygen pure. Hydrogen pure oxygen. And that hydrogen is used to reduce carbon dioxide. So now you think, why can't you make a catalyst which can decompose water? Right? Decompose water, get hydrogen. hydrogen can be used as a fuel. I told you carbon dioxide can be reduced to methane by a suitable catalyst, right? I have a lot of slides to show you. So that can be methane, which can be used as a fuel. So this solution, this problem of this energy materials is solved by chemistry, and engineering people help us to see how carbon dioxide can be efficiently reduced to methane, ethylene, like that. Okay. Nowadays people compete. There are a lot of industries which convert carbon dioxide. How do you have carbon dioxide? It's a pollutant, right? You burn petroleum, you get carbon dioxide. So carbon dioxide can be converted into useful products if you have good catalysts. So just see how people think about it. See, in this case, I have the American Chemical Society, magazine. Here, Chemical and Engineering News. A member of the American Chemical Society. It says talented 12. Every year, this chemistry and engineering news go to the scientists, young scientists in United States, and then see what is that they do. They are summarized here in this issue. Talented 12. Go and collect them, understand what they say. What is their research? One of the research is this: inventing catalyst for decomposing water. Inventing new energy materials. If you make hydrogen, how to store it? Nowadays in the US, you see, instead of petrol, you fill up petrol, you also fill up ethanol, C2H5OH. You also have alternative hydrogen fuel. You have a reactor where you can generate hydrogen, right? So future will not depend, should not depend on the petrol. It should be depending on newer energy materials. So chemistry comes to the help. How? You observe nature. You see how plants decompose water. Is it not interesting? And in your body, what happens? All the carbohydrates that we take up are burnt in mitochondria, in the cells. Burnt, oxygen is used, burnt, and you can generate energy. And what is the molecule which stores energy? You know how energy is stored in our body? ATP, phosphorus, you know more science than me. So, the energy is stored in the form of PO bond. That you learn from science. Energy is stored in the form of a PO bond. You cut it, you add water, you hydrolyze it, and you get the energy. Right? So, you learn a lot by observing science, understanding molecules. Right? Structure of the molecule. So, lot of scopes, I just, in one word I tell you what is going on in chemical science nowadays. So once you understand the structure of protein or DNA, then that means you have a new subject, that is structural biology. Once you have a good chemistry knowledge, go to the field of biology and then understand how hemoglobin takes up molecules, how DNA is important to us, right? That is how structural biology, now we say chemical biology. So the intention of chemistry is broad-based. It's now called chemical biology. What I do research to is new field, chemical biology. What is this? Use chemistry to understand biological molecules. As I said, hemoglobin, how does it function? Use chemistry. See, if you take hydrogen and oxygen, you say, right, H2O2, you combine them, react them, give H2O, right? Simple. One can collide with the other and reaction will happen. But now you take, just imagine, hemoglobin is very big like this building and oxygen looks like this. Whoa, whoa. How does it react with this building? See, it's a very big thing. The hemoglobin is very big and oxygen is very small. So how do they react? How do they interact? How to understand this? That is the problem now. So, dynamics. very large biopolymers like this, right? Protein like this. How do they interact with a very small molecule? So all your theories, chemical theories, will fail there. You have to have new theory, right? So two people will fight, we can understand who will win and who will lose. But when I go and hit the building, how to protect it? How to, predict the result, right? It's very difficult, right? So you have to have new theories. So that is how you say science is always expanding. So cell function, metabolism, everything, thought, how do we think, what is memory, what is its cell function, what is metabolism, all these can be answered only when you have a good chemistry. So but today, I took this particular aspect, molecules that change the world. Molecules that change the world. So when you think about chemistry, you should also notice that several new molecules have been made that change the world. That is the history of the science, not only the science, the art. How to make a molecule. It's not only science, it's an art. How to make a molecule. Whether go this way or that way, it's an art. People say synthesis of a new molecule is an art, and science of synthesis and its impact on society. So you should understand that. Then you understand the importance of science. We have several molecules, right? Molecules, what are the molecules? Every day, my own student, if you go to the laboratory, you'll be making 5 new molecules, not known so far. So millions of molecules are made. You try to define the structure. That's chemistry. Define the structure. And what I now talk about is molecules which have revolutionary effects, inseparable. They are very important to us. Revolutionary effects and become indispensable for human society. So several molecules have been discovered which facilitate our life in this world, right? And chemistry, as I said, should combine with biosciences, and so that we have an integrative science of life. So how much is the importance of science? Chemistry, that is why I said, is central science, right? So take a molecule like this. What is this molecule? Everywhere you go, in advertisements, they use this one. What is this? DNA. Good. Right? So that is the only important molecule which can change the world. What was changed? The structure. When they solve the structure, the people try to understand that, and nowadays we make use of it to design newer medicines. That's what I am doing today. So it's a very fascinating molecules. They are biological like molecules like DNA, and how to synthesize them, natural occurring molecules, how they are important in medicine, and blend all these fields, and you get newer fields and newer effects. So here I have noted down some of these molecules. One important molecule is here, aspirin. Some simple molecule, but it has changed our lifestyle, right? And you have hemine, right? Morphine. So it is interesting to see how these molecules were discovered, how they were synthesized, how they were artificially made in the chemistry laboratory. That's what I am telling you. So what is aspirin? You know that the universal synthesis. So I took a very simple example. Is analgesic, painkiller, right? Just read. It's also got acetyl salicylic acid. Salicylic acid, hydroxy, benzoic acid, and you have acetyl group here, acetyl salicylic acid. So this is used worldwide. You cannot live without aspirin. That's why it is a molecule which has changed our lifestyle, right? Previously, people used this willow bark. This contains aspirin. Not the aspirin. The derivative of acetyl salicylic acid is, this was used. It is present in the tree. People are using that. But later on, people found it's nothing but salicylic acid. 1874 itself. So now that molecule, that tablet is indispensable for us. We have to use aspirin, right? So it's a drug. This was the discovery. So once you know the history, then you will not forget about this particular aspirin molecule. You will not forget that. You start thinking about something, some other molecule which can it be used as in the place of aspirin. Like that, it makes you think. So once you know the history behind all these molecules, all these science. So this is about Felix Hoffmann. He only derivatized salicylic acid as base services. Okay? So how it is made? I think you know about that. It's not important for us. How important is how it is a painkiller, how it is synthesized, and how people have spent a lot of time in explaining how it kills pain. It's also known. The mechanism is also known, but we are not interested. But remember, a small molecule like this have changed our world. Another important molecule is vitamin B12. And you have heard about vitamin B12 already. What is that? It's a cobalt-containing complex. Where it is present in all these things. Eggs, meat, poultry, shellfish, milk, and all these things. Right? It's available in the market. This is the molecule. How do you know that this is the molecule? It's important for all these functions. I've tried to collect it. Right? And previously, before the discovery of vitamin B12, see, vitamin B12, just see how many Nobel Prizes have been given to one particular molecule. When you try to talk about, understand vitamin B12, you also develop science. You also develop a methodology to know its structure, X-ray structure, vitamin B. So this woman was given, Dorothy Crawford, was given Nobel Prize in 1964 for what? For determining the structure of vitamin B12. How much time she took? It's not small. Years, several years. But now, we have an X-ray machine in our chemistry department. If you get a good crystal of any new molecule, you put it in the machine, in 2 hours, you can get the structure. How atoms are arranged. How atoms are bonded. Okay? You can get it in 2 hours, 2.5 hours. It will give you automatically print it out. So much is development here to determine the structure of small molecules. You have a protein, single crystal of a protein, put it in the machine. It will give you the structure in a day or two also. Right? So, remember, discovering the structure of vitamin B12 involved development in science also. Right? Several principles were had to be developed to determine the X-ray structure. Right? So, this was discovered in 1934 and this was synthesized in the laboratory in 1965. Woodward synthesis in Harvard University. In Boston. It's there in Boston, Harvard University. So Woodward was given Nobel Prize for making the molecule. How many people are involved in making the molecule? How many days? How many years? It's not very simple. This laboratory, it's a very big lab. I visited Harvard and it is very interesting to visit. It will just look like a high school, but you will remember that you have entered into a big knowledge hub, Harvard University. And in 1981, he explained the function of vitamin B12. See, so many Nobel Prizes are there in one molecule. Now you will agree that this is the molecule which has changed our lifestyle. Right? How? See, before this vitamin B12, people suffered from anemia. They are lethal. You will easily die when you have anemia. Now that is treated by vitamin B12. He used an extract of liver extract which contained vitamin B12. He didn't know that. He used the extract to cure this anemia. From raw liver he extracted that. But then he isolated, later on people isolated and studied vitamin B12. They found that vitamin B12 is the intrinsic factor. So much is that the discovery of a small molecule, right? And the structure was determined, right, by Dorothy Hodgkin, and how nowadays people may extract it from liver and bottle it and give it to you, vitamin B12, right? I took a tablet in the morning also. So vitamin B12 has so much history, so much science, so much chemistry in it. Another molecule is heme. What is this? It is iron and a powerful organic molecule. This forms the basis of our blood. So we say iron is here, there is a vacancy here, there is a protein here, and you have a vacancy where oxygen can come and sit on iron. I am trying to simplify. So heme is another molecule, which changed the world. The understanding that this forms the basis of human blood or any blood. When you talk about that, you also see there are other types of blood. Blue bloods are there, which contain copper. There is also a green blood, right? So people think about oxygen storage here. So this is what I said, heme molecule. Okay? And that forms the basis here. This is the heme, your protein molecule. So you understand, you try to know the structure of the molecule. With the structure of the molecule, you understand how do they bind to molecular oxygen. There are 4 units, 1, 2, 3, 4. So when oxygen binds to hemoglobin, how many oxygen can be bound? 4 molecules. So it's very efficient. When hemoglobin goes from lungs to the tissue, taking oxygen, if it takes only 1 oxygen molecule, it's a waste. It's not efficient. So hemoglobin takes 4 molecules. Efficient binding. Remember, this oxygen is not oxidizing this molecule. So it binds reversibly. So much is there. So much principles are there. You can learn from each and every molecules that you come across. That's why we say the understanding of this molecule has changed the world. There's another important molecule. Taxol molecule. This is the best medicine, chemotherapeutic medicine for cancer. You have heard about cisplatin. That is the field I am working. Cisplatin, right? So, apart from cisplatin, Taxol is the most important compound for treating cancer. Efficient. Some people mix Taxol with cisplatin and give it to the patients. So Taxol is a naturally occurring molecule. And you see, Taxol is an anti-cancer natural product and is isolated from Barca Pacific UT. This molecule isolated. So it is available in very small quantities. So what should you do then? You should attempt to make it. Is it simple to make it? No. Look at this. This is the structure of the molecule. Taxol molecule. Very interesting. It's not simple. See, 6-membered ring, 9-membered ring, like that. So this structure of the molecule is very interesting. So how to make it? That itself is a challenge given to the chemists. How many people worked on it? Around 60 people. People who have taken PhDs, right? They go and assemble there, they are paid well, and they try to synthesize the molecule so that you can use it for curing your cancer, right? So, Nicolau, his group made this particular molecule. It's a challenge for chemists because it has a very difficult structure. 99 Nicolau. Who is Nicolau? Here is Nicolau. He only headed this particular discovery when making this particular molecule Nicolau. And he only wrote that book. I told you, Molecules That Changed the World. When I was traveling in Germany, I just came across this book, Molecules That Changed the World, written by this Nicolau. That's what I am talking to you. Right? so that you will understand the whole chemistry itself now. Once you talk about this particular molecule. So how many people were involved in this? It's a challenge. When you do that, you invent a lot of new reactions. You invent, when you just give new explanations, you give new mechanisms. How chemical bonds can be made. So one particular discovery, one particular challenge will just end up with newer methodologies, newer reactions, how to get them, how to use them. See? So a lot of people don't bother about that. Remember, making one protein molecule is not a simple thing. 60 postdoctoral fellows were involved in this. Some people, this is a summary of them, how they make it. Some people will be involved in this step, some three people. Some other people will be involved in making this particular reaction. Some other people in this reaction. So whole sets of people, they got involved in the synthesis. So much effort was put. Now we simply go and buy it in the market and use it. But so much efforts have been made, so much difficulties have been invested by people, chemists, to make this particular molecule. So now people understand the pharmacokinetics. How does it function? These are all things. So remember how this drug now acts in cancer. Now you should know, you attended the camp. Remember, it is anti-mitotic drug. What is mitosis? Cell division. Right? It acts at that point. When you have a cancer cell coming, growing, it goes and attacks that particular cell, asks some cell to die, you die there, commit suicide. This triggers that. This molecule triggers the cell to commit suicide. In nature itself there is a mechanism. Okay, apoptosis, cell suicide. There are triggers. This molecule triggers the DNA to die, to disintegrate. That is how it works. All drugs work like cancer drugs, they work like that. And you have a theory that whatever tablet you take, drug to take, they interact with protein or interact with DNA. That is what the science we try to do in our laboratory. So try to understand how it is metabolized, how it reacts, what are the side effects. All these are very important. It's an anti-mitotic drug. So this is what this another molecule. In fact, I made this slide in 2015. The Nobel Prize was given in physiology and medicine. Here is a molecule, right? For parasitic, including malaria and other things. This was made by 3 people. One is from China. This is the molecule. Another molecule, right? It's naturally occurring, and it was isolated. That was responsible for the 2015 Nobel Prize in Medicine, which acts against malaria. The pioneer is from a woman from China, right? So this is what it was a peroxide linkage. It's very unstable, peroxide linkage. But then they were able to isolate and use it as a Chinese traditional medicine. Okay. The next molecule I want to talk about is deoxyribonucleic acid, DNA. Right? This revolutionized science itself. Okay? I was benefited by this DNA molecule. Once you know the structure of the molecule, right, you enter into the field of biology and use the structure to solve the problems. That's what our students try to do in my laboratory. Structure of the DNA. So, it is called the molecule of life. Okay? It is a molecule of life. It's a genetic material for all cellular organisms. We are made up of cells, right? A cellular organism. So each and every cell contains this molecule. Right? So it's called a genetic material in cellular organisms. It's a gigantic molecule. We call it as a pig tail molecule. You notice pigs, your tail will be like this. So we call it always pig tail. You cannot control a pig, right? The tail also it always changes. So it's a pig tail molecule, gigantic molecule used to encode genetic information, to store the genetic information. So the DNA in our cells, so they have encoded the secret of telling, how many hands I should have, how many legs I should have, how many fingers I should have. All these informations are stored in the form of the structure of the DNA. That is why I say it is the molecule of life. This is a chemical. Simply it's a chemical. But that structure contains the message. What is the message? How many hands should you have? Should have. And if you take the apple seed, that just throw away. But that contains the DNA, which determines how the apple tree should grow. How many leaves should be there. What should be the structure? What is the color? All these are stored in the form of the structure of the DNA molecule. Right? That's why for all forms of life. And nowadays, for example, in 1990 or so, 89, we started this program in our laboratory. We just, I asked my student, one of my students, who is an associate professor in National College, I asked Dr. Indra to take up this molecule and see how it interacts with the copper ion. That's how we started. Now people know a lot of things about this molecule and how do they interact with metal ions. Right? One particular structure, if you know, see, chemistry is there about DNA, people talk about it, biology people talk about this, physics people, ethics is there, informatics is there, engineering is there, everybody talks about this particular molecule. In all advertisements, you see the structure of the DNA. So, it is the molecule of life. How do you say that? It is a structure. Okay? So, it is an informational molecule. How traits, characteristics of human beings are passed on from one generation to the other. That is in the form of the structure of the molecule. Okay? So, that particular structure of the DNA molecule is the DNA structure. If you have a structure like this, one particular amino acid will go here and stick. Another amino acid, another amino acid. When all the amino acids gather here, they form a bond, amide bond, and a protein is synthesized. So it dictates what amino acids should go here, what amino acids should go there. The structure, how this building is made, bricks. Just look at the shape of the brick. Look at the shape of the building, same. Right? That's how I say this DNA forms the brick. That is how we were built. So all these informations are there in the form of the structure of the DNA molecule. So you have new tabs. It doesn't do it immediately. It synthesizes RNA. That's called transcription, and it interacts with DNA. Metal ions help them. So a lot of things are there. So the structure of the molecule was determined in 1962. So these are the two people, Watson and Crick, Francis Crick. This Watson, Crick, these two people got Nobel Prize for this discovery, determining the structure. It's not overnight. They determined the structure first, then the structure, then the structure. They put all the informations together and then determined the structure of the DNA. It's not overnight, remember. They have tried throughout their life and assembled the structure. It's not very simple. In 1960, they got the Nobel Prize. In our Bharatdasan University, after 50 years, in 2002, we celebrated it. I brought together all the people interested in DNA and we had a celebration, we had a conference on it. Right? So, what did they say? Life is simply a matter of chemistry. What is life? It's a matter of chemistry. Right? I can predict after 5 generations how my son will look like. He will look like me. Right? There will be only small changes. Right? Because I know my DNA. If I know my DNA, if I also know, if I have a cancer, if I know my DNA, I can tailor a medicine to suit my own cancer. So much is the development. Personalized medicines. If you know the structure of the DNA. So, when the DNA, this man, Crick sold his medal, Nobel medal, to raise money by his own research. So 5 years back it happened. He sold it, the medal, for a big sum of money to foster his science. So if you know the structure of the DNA or any protein, any molecule, biological molecule which make up our body, for example, then I can enter into the field of biology, have a nice time. So for example, I started my research in my laboratory on DNA so many years back, 90 or so, 88, 90 or so, right? Now, how many people work on DNA in the same field? 30, 60 people work on it. Right? I'll also tell you from whom I took up the idea. Right? So, remember, the DNA structure, human genome, human DNA, structure was determined in 2001. And that led to another new world of science. You always think about the structure of the DNA and try to solve your problem, your own disease. Right? That's what an intelligent doctor does. Right? So know the structure of your own DNA and you can be, these are all about the DNA molecule, how they are present, how they are made up. See, this also makes up the DNA. And this DNA structure is made up of 4 letters, A, T, C, G, E, C, G. They form the basis. It's only 4 letters. Structure of 4 small molecules. They only make up this. What are these? It's here. That's good, you know about it. So adenine, guanine, cytosine, thymine. This is not there. Okay? So these 4 letters make up your DNA. Right? So this is the structure of the DNA. Right? You know now what is the technology? Suppose you have AT here, GC here, GC here, AT, they are all paired by hydrogen bonds. Right? So now people talk about, so your characteristics, your quality, how do you behavior is determined by the structure. Right? Now, a lot of people say you have some genetic diseases. You know why it is so? People say now it is because of the defect in the structure. So now you can repair the DNA. Okay? So you can try to edit the DNA. Take T. Suppose it is AT. It is now TG. Take A and then put G. Take G and then put A here. Editing, CRISPR technology. So other chemists are involved. One of the chemists here is using CRISPR technology, Chinese-based person. He will be the father of CRISPR technology. How to edit this? Okay, so you have genetic medicines. So remember, it is this structure of the DNA which determines all these things, right? So when you find out there is a mispairing, try to edit it. CRISPR technology. So DNA is small. You take a hair, cross section. You can also call this as a nanometer DNA, right? And what is the structure of the DNA? This is the normal structure. It is right-handed and it is optically active. You know what's optical activity? Not bother. So there is also, this is called pDNA. All life forms have this pDNA. All life forms. Take the mirror image of this one. This right-handed, see, comes from the screen, goes up, goes in, comes up. Right-handed. All our DNA is right-handed. What is this one? cDNA. It is the mirror image of this. Look at the DNA in this direction. See, you have a very good picture. Right? You draw a column in your house. and this is mirror image. So what is that my student did? You took up this one and interacted with copper compound, converted this into CDNA. It is very much unstable, this one, C conformation structure, not at all stable. It's not there in us, in this DNA. There's also one more, A-DNA. Take B-DNA, put it in water, and you need to stabilize the structure in sodium chloride. You have to have a lot of sodium chloride to stabilize this particular structure of the DNA. You know why do you take now sodium chloride? Stabilize the structure of DNA. Okay? Remove the sodium chloride, this will destroy, get destroyed. Structure will be disintegrating. Right? So this is another type of DNA. Why people are bothered about it? Because this should, if you take drugs, they interact with this medium. Sometimes it can be changed. So people are interested in some newer types of DNA structure. Look at what people do. When you say our DNA has stored the information, how many legs should we have, how should I behave, it's all there in DNA. Now you have a question. Can you use DNA in a computer to store the data? Look at that. Don't use your floppy disk here. Just throw it, it breaks. You want to have a good, cheaper, very cheap memory device. That is your DNA. See, look at that. DNA can be used in computers to store the data. So, the DNA, so we have been having the DNA for so many generations. Very stable structure. But all these disks are very unstable. So, people have looked at fossilized human beings' bodies, fossilized human beings. They have taken the DNA and analyzed it, and they found what is the language that person was talking 1,000 years back. Look at that. There are articles in Nature. You read that. No? You learn that. So, the language spoken by the man so many years back can be understood, unraveled, can be understood by analyzing his DNA. So, DNA stores, it's very stable DNA, that stores so much information can be stored in the form of DNA. This Goldman was able to store so much memory in 1 gram of DNA. He used 1 gram of DNA, can easily isolate it. He used 1 gram of DNA to so much memory is there. See, he says the cost of storage is rising, but our budgets are not. You need very big storage devices. You cannot go on making storage devices like floppy disks and other things. The CD drives, they are all very unstable. They will lose the memory soon. So DNA is the solution. See? And it uses, our computer uses what? 0, 1 binary. And DNA uses 3 numbers, 0, 1, 2. So, and they can be combined by AGC2 letters and encode the information into DNA. So, DNA can be now used as a storage. See, in 1 gram of DNA, the paper published by Watson Crick paper, that was stored, so many songs of Shakespeare were stored, and the Martin Luther King's speech about I Have a Dream, that was also stored in 1 gram of DNA. So much information stored in the form of DNA. Right? So chlorophyll, for example, it can be used as a switch in computers. So much is there when you observe nature, when you just look at them. So now we have DNA, use DNA to store and archive products of our own brain. We can store that and there is a twist in our brain. Here is again another Nobel Prize in 20, I think it's 16 or so. These people, what did they do? They got Nobel Prize because of DNA. DNA repair. So we are creating defective DNA, and that is also repaired by a mechanism which we have already. When that fails, we have cancer. So how to solve this? They found out how DNA can be repaired. Cisplatin we take as a medicine for cancer. That can repair the DNA. Defects in DNA and control cancer. So cancer is because of defects in DNA structure. When you are able to edit it or destroy the DNA, you can control cancer. And nowadays cancer is just like an epidemic. Everybody say, oh, cancer, cancer. So everybody should get together and find out a better medicine to treat cancer. So this is a structure and people have different things. Here is a lady who inspired me. Jacqueline Barton. In 95, I attended a conference in Germany. When she was giving a talk in a big hall like this, bigger than this, and I was standing at the corner. No place to sit. She was talking about all your discovery. Jacqueline Barton. She didn't get Nobel Prize, but she got the highest award or medal of American Chemical Society. Priestley medal. What did he do? When you take DNA, right? You take a compound like this. My students know how to make this molecule. Take this molecule and get the spectrum, emission spectrum. It emits light. It doesn't emit light in solution, aqueous solution. Okay, take a test tube, take the molecule, doesn't emit light. But you add DNA. Right? When you add DNA, the DNA interacts with the molecule here and then it starts emitting light. Look at that. It emits light. What does it say? It's a molecular light switch for DNA. It switches on light. It starts emitting light in the presence of DNA. That means you can use that molecule to detect DNA. A spectroscopy handle, a detector, an index, right? So it can detect DNA. So with DNA you have light, without DNA no light. These are the biggest discoveries he made. So she also discovered this is the molecule which we also made. We demonstrated in our own summer schools and winter schools we conducted. We draw scholars from all over India and we demonstrated, made this molecule and demonstrated how it emits light. There is another important thing. So when it emits light, okay, when you have another molecule here which can absorb the light, then what does it mean? The excited electron here, which is responsible for the color, it can travel through the DNA and get absorbed. So what is that she discovered? DNA can be used as a wire, molecular wire. So you discovered that the electron can be transferred through your. Just imagine this. See, she also got this Priestley medal for dedication to training young investigators. Right? So this is University of Berkeley, right? Okay. You just see a man, she's here, young lady. Do you know who is this one? Linus Pauling. See, people always say, people always I want to talk about it in the introduction. Why India is not getting a Nobel laureate? See, here you so if you go to one university, you see a lot of people concentrate. Not university, you get only 2 or 3 people doing science. When many people work in one place, right, you have motivation. So when I come here, I used to spend my time in the laboratory, right? I come here to talk to you. So when people interact, when the message is given to you, you are also empowered, you are also motivated. So all Nobel laureates are here. They talk. When people who work in science concentrate, you have more output. So in that respect, I should say, India is not a big country. It's a very small country. We are a very small country of science. That's why I told you, it is there in our constitution itself. This will be a science-based country. Right? You should propagate science. And here is an example where you should learn that it's not a small thing Nobel laureate. We try to copy what Westerners do here. Right? So we should have our own science development. So it should lead to that. Right? So remember, such small discoveries, not small, that's a discovery which becomes big. So DNA can conduct electrons through this wire. This is what we made. This is what our students made. We found that this can be used. This one, Dr. Rajendran in Central University, Tiruvallur, is an associate professor. We published a paper. We saw that this can be an anti-cancer agent. And this can interact with CSO. Barton herself crystallized this molecule in the presence of synthetic DNA. She saw that it interacts very well, closely with the DNA. So this, we said, we can be used as a drug for detecting the presence of DNA as a stain for dead cells. This is the molecule we found. People use organic molecule to detect the presence of dead cells. We saw that a ruthenium compound like this can be used to detect the DNA in dead cells. Here is, I said this is the talented people in 2017. Latest one. Every year I said. Here is a talented guy. In 2015 made this slide. So what is this? One of these 12 talented, what does he do? If you have a normal DNA, A, T, T, A, G, C, it's a normal DNA. It makes some proteins, makes RNA, and makes protein. How many? With 20 amino acids. The number of proteins is less. But look at that, what he did. He tries to engineer the DNA. Right? See? Instead of this one, he introduces, XY. A new base pair. Right? And then here a new one. He expanded DNA, engineered it. Engineering the DNA. With unnatural base pairs. What does he get? A different RNA. And then what does he get? Different 172 proteins. Right? So, when you engineer the DNA and try to CRISPR technology, use teach others different technique. Okay? So, he gets new proteins. So, innovative research. Try to play with your DNA structure, modify that, and get newer type of protein. So, the field is expanding. Here is another Nobel laureate whose work is based on this structure of the ribosome. This is ribosome. That's a laboratory where DNA is synthesized. That is a laboratory where a protein is synthesized. Translation. What is translated? The memory in DNA is translated. The storage is, the information stored here is translated into your protein structure. Okay. Right? So this is ribosome, the laboratory. So how it is made up? So a lot of people got together. One person analyzed, one group analyzed this structure, another one this portion. Ramakrishnan, he analyzed the structure of one particular portion, and all the informations were put together to get the structure of the ribosome. That got him the Nobel Prize. Why did they look at the structure of the DNA? What is the importance? Okay? So if you know the structure of the ribosome, you also know how to destroy it. If you have some insects, you know the structure of the ribosome. For example, any insect that you have, you know the ribosome structure. You can target the ribosome, destroy the ribosome. When you destroy the ribosome, the insect cannot make a protein. It cannot serve it. Right? So, remember, determining the structure of ribosome is so much important. Very complex laboratory where protein is synthesized and DNA is synthesized. And for example, here I have seen bacteria. Bacterial ribosomes block the function of the bacteria. So this discovery has lots of importance. Remember that. So Ramakrishnan got the prize, Nobel Prize. And you should also remember, he is the president of the Royal Society of Science in England. Okay? I think his father was a professor of biology in the University of Baroda, right? He was his son, and he went with BSc to UK, and the program, he was working in the program of determining the structure of ribosome. It's a protein factory, as I put it here. So it is not also overnight. All the people determine success in small things, put them together. So here is a scientist, again. So I said, molecules, they change the world. And here is, I'm just listing out people, scientists who change the world. Right? So what is after all the history? If you think about the history of India, history of the world. Right? Think about World War. Who comes to your mind? Winston Churchill. So the history of the world is nothing but history of these big people. History of science. It's not only just this thing, but history of these people, Einstein. Right? So this is Ahmed's wife. He also got the Priestley Medal, the highest award of the American Chemical Society. He passed away last summer, 70 years old. He won the Nobel Prize in 1999. For what? Femtosecond spectroscopy. If you have a reaction which happens within 10 power minus 15, 10 power minus 15 seconds, not in 1 second, 10 power minus 15 seconds, if you have a reaction, that can be detected by his technique. Femtosecond spectroscopy. Priestley, he was also a Priestley medalist. Right? So his discovery is not so Egyptian. Just see. He wrote in his room as Dr. Ahmed, even when he was 12 in Egypt. So he should also aim at that. Even now you should aim at the bigger thing, to do science, enjoy science, right? And this is his father, like that. So Dr. Ahmed, he wrote it in the bedroom itself, Dr. Ahmed, like that. And his service is not only that. He was also a member of the delegation to Egypt. Egypt had a problem a few years back. The revolution was there. And he was sent by Obama to Cairo. Right? He had dual citizenship, Egyptian as well as American. He was there to the demonstrators and pacified these people and to form a new government. So much is the influence of a Nobel laureate. And in fact, he used to travel in Middle East, right? And all the Middle East country people, they asked him, what is the secret to get a Nobel Prize? They asked him, what is the, he's a ambassador of science, peace to Obama. So what is the answer? His answer? How to get a Nobel Prize, even in India. What is the secret? He said, empower people like you, youngsters, invest in people like you. Now you understand the very basis of the inspired. So he said, take youngsters, very big things are done only when in youth. I can only guide my student, right? But my student carried out all the suggestions like that. They made a lot of molecules. So it all is there in you only, youngsters. I always say, 12-year-old girl is appointed in some IT industry in the US, right? I was reading in the newspaper. So 12-year-old girl, right? She is employed now in an IT industry, IT company in the US. She knows so much. I just forgot her name. A week back I read in the newspapers. So much is there, potential is there in you. You are capable, but you have to provide the chance, the opportunity to do good science. So you should be motivated. This again 2017 Nobel Prize last year. You know what did they do? These are the people involved. What did they do? Again, structure of the molecules. So with other techniques, you can know the structure but not deep. You don't know what is going on here. Resolution of the molecule. At bigger accident, we don't bother for that. But when you use this particular technique they discovered, you can determine the structure in pinpoint the molecule, atoms here. This is what the resolution. What is this technique they discovered? When you know the structure of the molecule which is moving in your body, you can solve all your health problems. It's not a very small discovery, right? This discovery will lead to solving all your medical problems. And people who make molecules as drugs, they will be benefited by this. If you know the structure, if you know the defect here, you can have a medicine for this. So in that case, this particular technique discovered by them, what is that? Cryo-electron microscopy. What is cryo? Very low temperature. What is liquid nitrogen temperature? 77 degrees Kelvin, minus 80 or so. I don't know exactly anyway. So you try to solve the structure of the Zika virus by this technique, cryo-electron microscopy. What do you do? Take the molecule at very low temperatures, freeze the molecule, okay, very low temperature, and then you use electrons. Electrons, collide the molecule and come out. You analyze the electrons, and then you get the structure. Similar to X-ray. X-ray strikes the crystal, the result comes, you capture them, analyze them, you get the structure. Same thing, this is an electron microscopy at very low temperature. Cryo-electron microscopy. 3D structure can be had, and biological process can be filmed, right? And all such things are possible with this cryo-electron microscopy. This involves a lot of mathematics also, a lot of modeling, not a simple one. So cryo-electron microscopy, for which the Nobel Prize was given in 2017. So I think I should talk about my own field. So lung cancer is the most common cancer in men. Breast cancer is the most common one in women. Several forms are there, and I said, World Health Organization says it is going to be in 2030, it is going to be epidemic. 30 million people will die out of cancer. Okay? So we know that it is because of cell division I told you. When you have a wrong DNA, wrong cell, that will lead to cancer. So you have to stop at the mitosis process. And you have drugs, as I said, and this type of cancer originates from defects in the structure of the DNA. And we say, cisplatin, how does it act? It binds to DNA like this and then destroys the DNA also. Now it's a textbook material for my MSc students to know about how does cisplatin, this molecule, interact with the DNA. So, how does it do? Goes and binds to DNA, changes the structure like this and destroys it. This is the theory. So what did we do? This is Dr. Rajendran from my laboratory. So what did he do? He made a very small molecule containing copper. Copper is there in green plants. What does it do? It transfers electrons. Transports electrons. That's what we study in our laboratory. So he made a small molecule like this, determined its structure, and he saw that it can cleave the DNA. When it cleaves the DNA, it can be also acting as an anti-cancer drug. So what started with DNA structure ends up in discovering newer molecules which can bind to DNA, right, and destroys that. So we have a lot of studies we saw that our molecules can have some values, minimum concentration of the drug needed to kill cancer cells. That is called ICP. Don't bother about this. The number is here, 0.29 like that. Whereas cisplatin has a higher number. Very small amount of our drug is sufficient, but we have not worked on that. We also saw that the cells can be divided like this. Mitosis. And my own colleague, Professor Kosa, the chemistry in the biology department, he helped us to determine structure C, apoptosis, mitosis, due to our drug, right? So that is what we have to show. So that's what we made long back, and that's how we take a small molecule amino acids also with copper, and they can also be good drugs. Anyway, we have now I've now told you about a lot of chemists also, and probably go faster, right? I want to tell you a lot of chemists, don't bother with that. I'll go to the end. So same person who discovered made the taxol molecule, at the end of the book he has a poem, Ithaka. Ithaka is a Greek poem. So science. Ithaka means imaginary world, imaginary island. You want to discover that. That is science. Imagine that as a science. So when you set out on your journey to Ithaka, right? Pray that the road is long. Your journey in science, let it be long, let it not be short. So, journey in science will be full of adventure, full of knowledge. Right? You will come across difficulties. You will come across very bad times. You will survive that, and then your thoughts will remain lofty to do science. That will remain lofty, and you will come across the fierce mastodons, dangerous animals, dangerous islands, and still, though you have difficulty in science, doing science, I have difficulties, but then I have enjoyed talking to scientists in Germany, scientists in Japan, and I have a wonderful time over here, okay? And then you should notice that the road to science, good science is long. Remember, at the end of the journey, you will have pressure on the way, summer mornings, joy is there. Keep Ithaka in your mind, do science. And then, arrive there is your ultimate goal. And that is not, you will realize, is the goal. Is the goal. But doing science, this journey itself is the real thing you wanted. So, arrive there is your ultimate goal. Let it last for many years. And what ultimately given the journey to science has given is, it has given you a beautiful body. Okay? You come across people who do good science, enjoy doing science, and remember that don't end up, let your journey not be, and when you find ultimately that Ithaka, science, you find that poor, and it has not deceived you. Science will not deceive you, but it has made you active, and so much experience you must already have understood what Ithaka means. So what is science? Not only that, you do science, your goal is science, but your journey of science is never ending. Right? Make it, take up the journey to Ithaka science, and you will have a good one. Remember, always the air is key. Curiosity is the key for explorations. And ultimately you will see, if you are wise enough, you will interact with wise people and do a better science. So collaboration is the underscore of all good science. That is what I have experienced. You should interact with other scientists. I have collaborated with a lot of scientists in Switzerland, in Japan, US, and I ended up with good science. So make science, Ithaka, your journey. That should be your goal, and that will not cheat you, right? You will have a good journey in science. I wish that you enjoy doing science, and I thank you for your patience.
is open for discussion. Any questions? Please be silent.
What is more important? Sometimes I ask the person, write the question on some papers, first time. So this took some time, right? 5 minutes time. Take 30 questions. That is the most important thing. I told you my own journey, right? And if you have some questions, you should come forward. The caste of the people, 471 marks, right? Look at that. You're not distilled people, no? You have the opportunity to come here. So I always talk about quantum numbers. How many quantum numbers are there? That is the problem. That's why I always tell the textbook is wrong. It's not the only thing. Even if you go to a professor in my own university, he will also say 4. So for so many years, I met the person who wrote the textbook, that Programe. I told him this is wrong. Right? No, I was talking to the KV school teachers in IIT Bombay. We had a reference course for the KV school chemistry teachers. I was addressing them like this. I asked the same question. They also said 4. 50 years old, 55 years old teachers. They have taught chemistry for so many years. They also had the same answer. 4. It's not 4. When you talk about spin quantum number, I say it is half. When you put it in a magnetic field, it assumes 2 values, plus or minus. So 5 quantum numbers. Really, I want to look at the recent. We are going to have a new textbook, and this is how the science books written are not good. They should teach you with all these histories. Or your teachers should be able to do that. Not from the point of view of examination. Right? How about you? You are from which school? Oh, same, Saudambia. Oh, this uniform. Oh, right. What is your aim? What is your aim? You want to become a doctor? I don't find a problem with you. Though I talked about science now. The doctors also applied science, right? So don't bother about it. You just take it up. But such a camp would encourage you to talk about your medicine, everything in terms of molecular mechanisms. These molecules. How about you? Same school? Same school, right? What is your aim? IIT, good. See, this is IIT. In Mumbai IIT, where I was teaching people like you for the experiment. Even if they come late by 5 minutes, we will decrease the points by 0.5. Marks will decrease 0.5. They are very active people. When they come into the lab, they will be at it, at the laboratory. Each and everything carries marks. And the type of people who come to IIT Mumbai, the youngsters, they are all different. Right? They are all different really. The very atmosphere in IIT-B is different, exactly different. You have an infinity corridor. You can walk along that. It's a very good name. Infinity corridor. If you go that, the growth is infinity, like that. All the laboratories are built along this. So students are very active guys. Every day you will have some meetings, some lectures. They are brought up like that. They are made active. It will be good. I know very well. How the IITians are different. They go there, live there. They imbibe a lot of qualities. They know how to talk to me. They know how to interact with me, try to know latest science, latest physics, latest engineering, everything. Good, all the best. How about you? What would you like to become? Science is good. What science? I should ask this question after the camp is over. Anyway, yes, you want to say something? Answer the questions. A lot of questions are there. You like physics? So where Abdul Kalam, what is the Abdul Kalam study? Where did he study? Abdul Kalam. St. Joseph's College. BSc Physics. BSc Physics. So then he went to MIT in Chennai to learn get B.Tech. So there are a lot of things about teaching. A lot of things about teaching. No? This person began to inspire Dr. Ramasamy, I told you. He was also a PUC from St. Joseph's College. He used to visit us often. I think I should get some time to bring you for the next camp and talk. He was the secretary of the Department of Science and Technology. Right? Okay.
On behalf of participants, I would like to extend my heartfelt thanks to you, sir, for your informative lecture about molecular chemical science. Thank you very much, sir.